Let's take a dive into the world of genetics with its breathtaking possibilities. Now, take a guess. Which of these animals are we humans closest to genetically? Monkey, tiger, mouse, or a lion? Unfortunately, not a lion. We are 85% genetically related with the mouse and share 99% of our DNA with monkeys. Now, some history of genetics. The story of the father of genetics, Gregor Mendel, is certainly tragic. He was an Austrian monk who had a way with plants. He grew hybrids of pea plants and meticulously recorded which traits were passed on to the next generation of plants. This eye for detail and tenderness allowed Mendel to grow several generations in this manner. In 1866, he published a paper in an obscure journal in which he stated the 3 laws of heredity. You would expect that this launched the science of genetics, but it was forgotten for 40 years. Eventually, it was simultaneously rediscovered by 3 scientists in 1906. This was the year many science historians say genetics was born. Charles Darwin, although you may not realize it, made several contributions to the budding science of genetics in 1859. Surprisingly, he did this without any knowledge of genetics. On the Galapagos Islands, he noticed that there were 13 finches of the same group, but they didn't have much of a resemblance. He hypothesized that they came from a common ancestor and branched off according to what was the best trait in their island. This was called the survival of the fittest. The best and necessary genes were selected for and survived to pass on their traits. In his own way, he predicted mutation, inheritance, and genetic drift. Scientists today find and deduce common ancestry based on shared genetic material. It is amazing how Darwin did this without knowledge of heredity. The most famous people in the history of genetics are James Watson, Francis Crick, and Rosalind Franklin. Franklin has been called the Dark Lady of DNA. Watson and Crick are almost always credited as the discoverers of the double helix of DNA in 1953. However, Franklin played an often unnoticed role. She took an X-ray photo of DNA that made it very clear if it was a double or a triple helix. Interestingly enough, Watson and Crick were shown that picture by chance. That helped solidify the results, but she never got the credit until recently. Her male colleagues sidelined her, and this might happen in scientific research even today. Genes are transcribed into an intermediate called RNA, which stands for ribonucleic acid. It just is an extra oxygen compared to DNA and one substituted compound. RNA is translated in a cellular machine called the ribosome to form a protein. It's not that this DNA is junk. It just doesn't code for proteins. Research is being done to determine what it actually does. It probably plays an important role that is yet to be discovered. 98% of our DNA is junk DNA. The Human Genome Project is an international attempt to come up with a reference genome for humans. We have 6 billion of those building blocks altogether, so it certainly is an enormous task. Gene sequencing machines were used to do this by decoding fragments and overlapping them to form a genome. Crispr is the newest gene editing technology, only introduced in 2012. Surprisingly, it's a bacterial defense system. Cas9 is the enzyme that cuts the gene, and it is replaced with the correct gene or deleted. This is applicable for any sequence. It is 6 times more efficient and accurate. Genetic screening and gene editing are risky, but do have some positives. We can prevent lethal diseases, determine ancestry, and based on our mutated genes, determine our possibility of getting a disease later on. There are several negative ethical implications, however. We could select for traits not essential to health and create designer babies. For example, like athletic ability. This could mean that people with those undesirable traits could be considered to have worthless lives. We could also ignore our potential to change and adapt as humans. Could also promote social inequality. There are many consequences of possible gene editing. Just imagine the secrets that can lie in our DNA, and we can all have a part in unraveling it.